experimental proof also. Like, initially what we have done, PSA and Histones are pre-incubated for some time. Then platelets were exposed to that combination, and we observed sudden significant reduction in the platelet aggregation. We found similar trend in case of neuraminidase treatment also. We got same type of results in case of viability. This clearly suggests histones are interacting with sialic acid content of TLR2 and 4 receptors. So the observation, what we observed from up to this means when extracellular histones are exposed to platelet, they directly interact with TLR2 and TLR4, or they can directly interact with other cell surface receptors. That leads to increase in the intracellular calcium and reactive oxygen species within the platelet, and that leads to either death or activation. At lower concentration, platelets are undergoing activation. At higher concentration, platelets are undergoing death. If so, what kind of death it is? So we ask next question, what kind of death it is? There are 10 different types of cell death mechanism. Out of that, these 4, apoptosis, necroptosis, and autophagy, as well as ferroptosis, are well understood in case of platelet. So we were interested to know what kind of cell death is happening in presence of histones. So in order to confirm this, what we have done, we used inhibitors of different cell death. So for example, FS1 and LPX1, these two are inhibitors of ferroptosis. NSA and NS1, these two are inhibitors of necroptosis. ZVAD FMK, it is a pan-caspase inhibitor, and we can consider this as an inhibitor of apoptosis. Ammonium chloride and 3-MA are the inhibitors of autophagy, and we used H2O, hydrogen peroxide, as a positive control. From this data, we came to know that only ammonium chloride and 3-MA protected the cell from histone-induced toxicity. From this data, we can say autophagy is the death mechanism is happening in case of histone treatment. So, if so, we need to confirm with the markers. For that, we did immunoblotting, and we assessed the markers of autophagy like Becklin-1, LC3, ATG12, ATG5, and ATG16L, and beta-actin served as a loading control. It clearly confirmed that autophagy is the type of cell death is happening in case of histone treatment. Then, in order to know the molecular signaling, what we did initially, we used different inhibitors like salubrinol. Salubrinol is a ER stress inhibitor. Enjostarin is a protein kinase C inhibitor. When intracellular calcium level increases, then only protein kinase C get activated. Then Bapta-AM is a cell-permeant calcium chelator. If there is increased levels of intracellular calcium, it can chelate the intracellular calcium, so that there won't be any sort of cell death. And meantime we used mitotempo. It serves as a mitochondrial targeted antioxidant. We didn't observe any sort of impact on cell viability by mitotempo. It clearly suggests there is no mitochondrial stress in extracellular histone treated platelets. Whereas in case of salubrinol, we observed significant increase in the cell viability, as well as in enjostarin and Bapta-AM, as well as calpain inhibitor. Calpain inhibitor also, calpain is a enzyme protease which are calcium dependent, and earlier data what I have shown, from that we can say there is a significant increase in the intracellular calcium. In order to confirm once again, what we did, we assessed the levels of intracellular calcium with the increasing concentration of histone, and we found dose dependent increase in the intracellular calcium. When there is a ER stress, then only we can expect intracellular calcium, rise in the intracellular calcium. So we interested to know whether ER stress is happening our ER stress is induced by extracellular histone or not. We assessed markers for endoplasmic reticulum stress, and we observed there is a increased expression of markers related to ER stress. And also meantime, by inhibitors also we confirmed that there is a ER stress. From this data we can say extracellular histones are inducing ER stress, or we can say extracellular histones are inducing autophagic death via ER stress, mediated through ER stress. When ER stress, endoplasmic reticulum stress happens, there will be accumulation of unfolded proteins, because ER is the major site for post-translational modification. If something happens, something goes bad, those proteins are targeted to proteasome. So those proteins are useless protein, unfolded proteins. They are undergo polyubiquitination and targeted to proteasome. In order to confirm whether unfolded protein level get increases or not, we did Western blot by using polyubiquitination antibodies, and we found dose dependent increase in the levels of polyubiquitinated proteins in the platelets treated with different concentration of histones, and we used Mg132 as a inhibitor of proteasome. When proteasome get inhibited in the platelet, the levels of unfolded protein get increases, which are ubiquitinated protein level get increases. This data clearly suggests extracellular histones are inducing ER stress, thereby unfolded protein level get increases, and meantime autophagic death is happening. So what we observed is when platelets are treated with histone interact with sialic acid content of TLR2 and TLR4. That leads to ER stress. When ER stress happens, intercellular calcium level get increases, and meantime phosphorylation of elongation factor happens, as well as phosphorylation of FERC, that in turn phosphorylate initiation factor 2 alpha. That these two events finally leads to autophagy. When meantime there is an inhibition of proteasomal activity. So because of that reason, in a cell we can expect accumulation of unfolded and misfolded protein aggregates, specifically ubiquitinated protein level get increases, that leads to death. So once we know the molecular mechanism of extracellular histone induced platelet death and activation, we ask next question: do phytochemicals provide protection against histone induced toxicity? Because there we didn't find any sort of literature or information on the usage of phytochemicals against histone induced toxicity. So based on these facts, we screened around so many phytochemicals. In this only we have given 9 or 10 different phytochemicals, redox modulators, and antioxidants. For example, berberine, curcumin, rutin, crocin, urolithin, celestrol, quercetin. These are the phytochemicals. Melatonin serves as a redox modulator, and NAC serves a NAC is a antioxidant. Uric acid is a metabolite. Among these, only quercetin protected the platelets from histone-induced toxicity. And we observed whether quercetin inhibit platelet aggregation induced by histone or not. It significantly increases, it significantly decreases the platelet aggregation induced by histone. And meantime, activation markers also significantly decreased in presence of histone, and proteasomal activity significantly restored back to the normal in presence of quercetin. This data clearly suggests quercetin is preventing histone-induced toxicity. In order to confirm further, we did confocal imaging of platelet in presence of different inhibitors as well as quercetin. And you can see in presence of quercetin, there will be a significant restoration of platelet function. In case of salubrin also, as well as ammonium chloride and ammonium chloride also. These two are inhibitors of autophagy. So in case of histone alone, there is a significant change in the morphology of platelets. This data clearly suggests quercetin is curbing histone-induced toxicity towards platelet. So these are the data related to in vitro. Then we did 3 different types of animal experiment. First animal experiment, the down panel or below panel. We use 2 groups of animals. One is quercetin treated animal, another one is PBS treated animals. What we did, we gave quercetin every day, 50 mg per kg body weight every day for 5 days. And in another group, we didn't give anything. We give only PBS, same volume of PBS. And after 5th day, what we have done, we sacrificed the animal, and we isolated the platelets, and we did aggregation of platelets, platelets from quercetin treated animals and PBS treated animals, and we performed platelet aggregation in presence of histone. In case of quercetin treated platelets, when we add histone, these platelets are not primed platelets, and aggregation was very low when compared to PBS treated animals. This clearly suggests priming of platelets with quercetin before the exposure of histone prevents aggregation to some extent. And we assessed the LDH level release assay in the platelets in presence of histone. Here, the platelets from PBS treated animals are more sensitive to the histone when compared to platelets from quercetin treated animals. Same trend was observed in case of aggregation. This data clearly suggests it is better to prime the platelets by quercetin. Then we did second experiment, that is top panel. So what we have done in that group, we used 4 groups with 5 animal in each group. First group receives vehicle, that is PBS, up to 5 days. Then second group receives histone alone through IV. Third group receives quercetin orally from 0 to 5 days. And on 5th day, we gave histone chart through IV intravenous. Fourth group, we gave only quercetin orally for 5 days, 50 mg per kg body weight concentration of quercetin. Then we sacrificed the animals and isolated the platelet. And in presence of ADP, we observed the aggregation. Here you can see in case of fourth one, the aggregation is very low. Aggregation percentage is very low. In case of group 3, there is a significant reduction in the aggregation compared to group 2. Group 2 is histone alone. This data clearly says that it is better to provide quercetin before histone treatment, like that. So we assessed some markers related to autophagy, and we observed low levels of autophagy markers in case of 3 and 4 compared to 2. Aggregation data also significantly decreased in the 3 quercetin treated group. And we have taken lungs image, macroscopic image as well as microscopic image. In microscopic image, one can observe this intactness of lung structure in group 1 as well as group 4. In case of group 2, there is a accumulation of inflammatory cells all around the lung. In group 3, there is a significant decrease in the accumulation of inflammatory cells, and clear indication of alveolar sac in the lungs treated with animals treated with quercetin followed by histone. So then the third animal experiment what we have done, it is a pulmonary thromboembolism in vivo. In this experiment, generally what we do means we give epinephrine and collagen through IV when we provide epinephrine and collagen through intravenously, platelets which are present in the circulation get activated. Means when they get exposed to collagen and epinephrine, platelet get activated and they form a thrombi. Those thrombis will travel to the lungs and form a embolism. So within 20 minutes, animal will die. This experiment, this model is pulmonary thromboembolism. So what we have done, we used around 6 groups of animal. Each group consists of 5 animals. In first group, we gave a vehicle up to 5 days. On 6th day experiment. Second group, up to 5 group vehicle, that is PBS. In 6th day, what we have given, collagen and epinephrine, we provided to the animal, and animal will die within 20. We observed the time of death of an animal. And in third group, what we have done, in 5th day, we injected histone through IV and we allowed animals to stay for 2 hours, then followed by collagen and epinephrine. Already histones are, in this case, in third group, histones, once we inject histones to the animal through intravenously, they may activate the platelets. In other words, you can say platelets are already primed to undergo activation because of presence of histones. During such situation, after 2 hours, if we give collagen and epinephrine, we can expect activation, robust activation and formation of thrombi will be more in this case. In group 4, what we did, we up to 5 days we gave PBS. In 5th day, quercetin and histones are pre-incubated, and that pre-incubated histone quercetin was given to the animal and allowed animal to stay for 2 hours, followed by the injection of collagen and epinephrine, in order to know whether quercetin prevent the priming of platelet before giving collagen and epinephrine. That was our idea. In fifth group, what we did, up to 5 days, every day we gave quercetin 50 mg per kg body orally. On 5th day, we gave a histone shot, 50 histone shot through intravenous, and allowed animals to stay for 2 hours, followed by collagen and epinephrine. In this case, we are already priming platelets with quercetin, followed by histone, and followed by collagen and epinephrine. Sixth group receives only quercetin, works as a negative control. So in this case, if you observe the lungs in first group looks intact and alveolar sac looks intact, whereas in second group, it is collagen and epinephrine treated group. There is a lot of necrosis or thrombi formation. You can observe microscopic image. You can observe a lot of thrombi, accumulation of microthrombis or more. In case of third, the number of microthrombis is significantly higher when compared to group 2. Okay, in group 3, number of microthrombis is significantly higher when compared to group 2. In fourth group, this one is pre-incubated group, both quercetin and histone pre-incubated, followed by epinephrine. It looks similar to group 2. Whereas in case of group 5, here you can see significant reduction in the microthrombi when compared to group 2, group 3, and group 4. So this clearly suggests it is better to give quercetin before collagen. Why we have done all these experiments means, sepsis. Sepsis is a deadly clinical condition. We can classify sepsis into 4 stages. One is SIRS, that is systemic inflammatory response syndrome. Second one is sepsis. Third stage is severe sepsis. Fourth stage is septic shock. Till date, there are so many clinical data suggest that approximately 20 to 35% of patients with severe sepsis and 40 to 60% of patients with septic shock will die within the 13 days. 30 days, within the 30 days. Means these are not reversible clinical conditions. These are irreversible clinical conditions. One patient's enters into severe sepsis stage or septic shock, it is very difficult to bring back that patient to normal condition. Whereas if patient is suffering from sepsis or SIRS, then there will be a great chance to bring back that patient to normal condition. So we are thinking, by our experiment, if we provide quercetin either at SIRS stage or sepsis stage, we can bring back the patient from that deadly condition to normal condition. It takes long time to do that, but our animal preclinical data suggests that it is better to give quercetin before the onset of severe sepsis or septic shock. So these are some, clinical markers. By using this, we can categorize the patient who are suffering from SIRS or sepsis or severe sepsis or septic shock. With this, I can conclude, like during severe microbial infection, the upsurge of histones and its toxic effects on platelets has clinical implications. High levels of histones and platelet activation are linked to both localized and systemic infections in sepsis. Quercetin, to combat the damaging interaction of histones with platelet toll-like receptors, could possibly serve as a potential treatment approach during sepsis. Thank you, one and all, for your patient hearing. Meantime, I thank my mentor, Professor K. Kemparaj sir, University of Mysore, for his continuous support and encouragement during this entire study. And I thank DST sir for funding. And I thank Professor Mugesh, is a collaborator from IASC. Entire data done by Manikanta, Mr. Manikanta, with the help of Dr. Naveen and M. Shekhar. Thank you, one and all. I'm happy to take questions. Thank you.
Okay, now the topic is open for discussion. So participants, if you have any doubts, you can clarify with Dr. Girish.
Thank you very much for the very good presentation, sir.
Thank you.
This is Navya from Department of Biotechnology, Downgrade. Sir, I have one question.
Yes,
what happens if the malfunctioning or malformation of the histone protein occurs, whether it can be corrected or not?
Madam, can you repeat the question? Sorry, can you repeat the question?
Sir, what happens if the malfunction or malformation of the histone protein occurs within the cell?
Malfunctioning of, madam?
Is malfunctioning or malformation within the histone protein structure.
Oh, okay, madam. That leads to that is a epigenetic modification. If acetylation and deacetylation, methylation, those things leads to activation of different genes, expression of different genes. That is epigenetic modification. What we are trying to say here is, when cells are undergoing death, cells release histones as a dam.
Actually, your topic has I have understand your topic,
yes,
and this is very nice piece of work. But my question is only with respect to the histone protein malfunctioning or malfunction.
Oh, yeah. Then there will be a continuous activation of particular gene or expression of particular gene that leads to different sort of problems. Epigenetic modification. Epigenetic modification happens.
Okay.
I hope I answered your question.
Yes.
Oh, thank you. Thank you.
Sir.
Any other participants? Yeah.
Sir, I'm Dibyendu Shekhar Mohanty. Sir, I have a question.
Yes.
How this histone protein is purified, and after that it is purified to what level during application?
There are some methods you can purify them, but it is difficult to purify in an individual form, like 2A, 2B, 3, H4. It is difficult. So because of that reason, they are using recombinant proteins for the studies and all. What we are used is a total histone. It includes all, and we don't know the percentage. What is the percentage of H1, H2A, H2B, H3, H4? We don't know, but it is a whole histone what we have used in our study. So if you want to use individual, it is better to take recombinant proteins. It is very difficult to isolate individually. You can isolate easily total histone proteins.
Yes, sir.
But it is very difficult to isolate individual histones because their molecular weight is narrow, and the composition is similar. So because of that reason, it is difficult to separate by using conventional chromatography techniques.
Thank you, sir.
Thank you.
Sir, may I ask?
Yeah, please, sir.
Sir, the questioning that compound is isolated from phytochemicals or synthetic chemicals?
Sir, I think we didn't isolate quercetin. We purchased it from company like Sigma. Like what we initially we were interested to know why people are not used phytochemical against histone-induced toxicity. So because of that reason, we screened around 20, 25 or 30 different phytochemicals, and we found only one, that is quercetin, is inhibiting histone-induced toxicity. None of the phytochemical other than quercetin did not protect the platelets from histone-induced toxicity.
Thank you, sir.
Thank you.
Sir, one more, sir.
Please, sir.
Sir, this is a basic question only. After the corona, many of the youngsters are getting cardiac arrest or heart failure or something like that.
Sir, it is it is difficult to say. There are so many controversies are there. Some people are telling vaccines are responsible for it. Some people are telling post-medicated complications and all. But there could be post-medicated problems. Still, scars are there in lungs. The patients who suffered from COVID-19, scar formation was more during COVID-19 infection. So still they are suffering from scars. And we cannot pinpoint to a point, like, this is the reason.
Okay.
But in COVID also, there will be a lot of surge in the extracellular histones in circulation. Like, that is one thing. And we found like continuous activation of platelets during COVID-19, and a lot of thrombi formation in and out, especially in lungs.
Thank you, sir.
Thank you.
Sir,
Yes, sir, please.
sir, I have one question, sir.
Yes, sir.
So I have read one research paper, sir.
Okay.
Some snake bites, that is poisonous snake biting,
Yes, sir.
increases platelet necrosis. Okay, sir.
Necrosis. Yes, sir. Necrosis.
Platelet necrosis.
Yes,
So it is a one reason, sir.
yes, sir.
So is there any reasons to histone toxicity by taking the common drugs, sir, like antipyretics, antibiotics, and other these common drugs they are taking? Is there any increase in the level of histone, sir, in the body?
Sir,
Or it is a type of side effects, I say.
Okay.
So that is a consumption of drugs, sir.
Yes, sir. Sir, what happens when you used more amount of drugs? When you use more amount of drugs, we can call them as xenobiotics.
Yes, sir.
When cells are get exposed to xenobiotics, cell may undergo death. So that death leads to release of DAMPs. One such DAMP is histone. But when histones are under normal condition, there are other proteins, what I said, natural molecules like C-reactive protein, heparin, suramin, albumin. They will quench the histone and they won't allow histone to induce toxicity. Whereas in case of infection, there are two things happens during infection. For example, if person is suffering from sepsis and you are loading with you are injecting so much of antibiotic, and we don't know whether that microbe is sensitive to this antibiotic or not, but we are loading antibiotic. So when you load antibiotic, and meantime, if your microbes are dying, that microbes should be eliminated or engulfed by our macrophages or phagocytic system. So if you use more amount of antibiotic, your phagocytic system will undergo will experience a lot of oxidative stress. Their activity goes down, their function goes down. So because of that reason, you can expect accumulation of dead microbes which are not cleared by your system. That dead microbe induces inflammation. We can call that as a sterile inflammation or pseudo inflammation. That leads to a lot of complications. So that's why it is better not to take much drugs, continuously. For based on prescription, it would be nice. In case of sepsis, what happens now, sir? The idea of the mindset of doctor is to protect the patient. So because of that reason, they will infuse more amount of antibiotic. But in the patient, phagocytic system is already undergoing a lot of stress. It cannot do its function. That leads to many complications. So it is that's why clinical data suggests more than 50% patients who are in septic shock or severe sepsis, they will die.
Yes, thank you, sir.
Thank you.
Hello,
Sir,
sir.
you have given a
Sir,
very nice lecture, sir. Thank you.
you, sir. Thank you, sir.
Hello, sir.
Sir, please, sir.
Hello, sir. Myself is Dr. Venkesh, Davangere University.
Yes, sir.
Sir, so all the subunits of histones can be separated in 2D, and by individual subunit can be used to study the stimulator or death of an platelets can be possible, sir.
Yes, we can do, sir. But the problem is, in 2D you can separate them, but what is the amount you will get it? That is
But lower concentration. Yes,
lower
yes,
concentration. That is
yeah,
not
yeah.
enough to do your work. So because of that reason, it is better to buy recombinant. We have recombinant histones. We have around 3. We have, but
Okay.
10 microgram, not 100 microgram of recombinant protein cost you around 17,000.
Okay.
100 micrograms.
Okay.
So
It's
we
costly.
need mgs wise. Yeah.
Yeah.
It become costly. So because of that reason, we use total histone.
Okay. Thank you, sir.
Thank you.
So if there are no questions, any one of the participants can propose vote of thanks to Dr. Girish.
Yes, sir. It is me from Gadag, Somappa B. Pillai, to thankful to the Sir K.S. Girish, sir, on the behalf of the University of Mysore and on the behalf of all the participants. I would like to give heartfelt thanks to Professor Girish, sir. Thank you, sir. Thank you very much, sir.
Thank you.
For giving such a wonderful and highly informative lecture, sir. Thank you, sir.
Thank you, sir. Thank you. Thank you, sir. Rajkumar, sir.
Okay, thank you, Girish. I think the lecture given by Dr. Girish is very impressive because they themselves have worked and whatever the work they have carried out in their lab, he has shared that. That is very important when a platform like this actually. So we really appreciate Dr. Girish that we hope that in future he will come out with some good information which can cure some of the diseases which the people are facing across the world, around the world. Okay. So thank you, Girish.
Thank you,
wish
sir.
you all the best. Yeah.
Thank you.
On behalf of Academic Staff College and on behalf of University of Mysore, we immensely thank you.
Thanks, sir. Thank you, sir. Thanks for the opportunity. Thank you.
Okay, participants, we can you can take a break and we can assemble exactly around 11:40 back again.
Thank you, sir.
Thank you, sir.